Okay, good morning, everybody. First, I'd like to say thank you to the organizers for giving me this opportunity to speak. So I want to be talking to you today about some of the work we've been doing as part of the CLL pilot study of the 100,000 Genomes project run by Genomics England in the Shoe Lab in Oxford. I have nothing to disclose. So you've probably seen this slide already this morning, or at least this figure. First, I want to talk about what we already know in CLL just briefly. So we all know that CLL is an extremely heterogeneous disease in terms of disease course, progression, and genetics. One of the strongest markers we have for patient progression is the mutation status of the immunoglobulin heavy chain variable region. And we know that the mutated patients will do far better than the unmutated patients and present with less aggressive disease and a longer time to progression. What we also know about is TP53 mutations, where we know that patients presenting with a TP53 alteration will do far less well than patients without them, even after a course of chemoimmunotherapy. But first-line TP53 abnormalities only occur in 8 to 12% of patients at diagnosis, and 25% of CLL patients will still relapse within 2 years from first-line chemoimmunotherapy. So this means that over 50% of patients who are relapsing present with no known marker for refractoriness. So why do these patients progress? Well, we've had some interesting talks this morning that have shown a number of additional biomarkers that are starting to come out of genome and genomics work. We have identified a huge number of potential driver genes that we know in many cases have an effect on prognosis. These include TP53, we've already talked about, SF3B1, MYD88, Notch1, IKZF3, and so many more, with a huge tail end of rarer mutations. But of course, we can't use all of this in the clinic. Currently, 17p deletion, TP53 mutation, and IGHV status are the three main markers that are used in the clinic at the current time, if that. And unfortunately, these three markers have to be identified using multiple techniques. So we have FISH, array karyotyping, microarrays and targeted panels, and Sanger sequencing for the IGHV. So the ideal here would be to be able to do this in just one single technology, and WGS is the thing that I believe will allow us to do that. So we've talked about existing biomarkers. Whole genome sequencing gives us the opportunity to do biomarker discovery, particularly in the non-coding space, which we know makes up over 98% of the human genome. Now, there have been some whole genome studies already carried out in CLL. So far, we have 236 whole genomes, with more coming every day. Many of these studies have focused on good prognosis CLL patients. Not so many have been carried out on relapse refractory patients, and we also don't have so many on patients that have been included in clinical trials. So as part of the Genomics England 100,000 Genomes project, so far we've sequenced 488 patients from clinical trials. Now, these patients come from 6 different UK clinical trials. That's CLL210, CLL Clear, Admire, Arctic, Flare, and Rialto. And these trials cover the whole spectrum of disease, from the Clear patients who are untreated at diagnosis to the CLL210s who are relapse refractory, and the remainder who are frontline patients. So these are just a few of the initial results that we've been seeing. So we're able to identify driver genes that are mutated across all of these cohorts that we have. We find SF3B1 mutated in over 17% of our patients, Notch1 in around 15%, TP53 in a little over 12% of our patients. We also find an enrichment for TP53 in our relapse refractory patients, which is what we expect to see. And these mutations that we're talking about here are somatic alterations. We've also combined a number of different pieces of software which are designed to look for significantly mutated genes in the hopes of identifying potential new driver candidates. So here we've combined MUT6CV, Oncodrive FML, Oncodrive Clustal, and DNDS-CV, and we've come up with two potential genes of interest, WDR74 and IRF2-BP2, that are worth following up. So now I want to talk to you about something that we can only do using the whole genome, or at least some work that we've been doing that has harnessed the whole genomes that we have. So firstly, we've been looking at genomic complexity. Now here we're using a simple measure of genomic complexity. Less than 4 CNAs is not complex. More than or equal to 4 copy number alterations is what we are calling complex here. And we can see a significant effect of genomic complexity whereby the patients that have complex karyotype do less well. Interestingly, we also see a strong, if not stronger, effect of the total length of those copy number alterations, whereby patients that have less than 15 megabases worth of total copy number alteration do better than those with more. Now, at the moment, 15 megabases is simply the average, so it's above or below the average of our cohort, but we are working on methods to make that more scientific. Something else that's interesting is by looking at every single mutation in the entire somatically mutated genome, we can identify signatures in the DNA that are related to different mutational processes. So this work was pioneered by Alexandrov in 2013, and we have selected signature 9, which is AID-related enzyme activity, which we have tried to use as a proxy for the IGHV mutation status as derived by Sanger. Now, out of 97 of our patients, only 8 disagreed between the Sanger and the signature 9. And when looking at our patients in a wider group, you can see that we get clear separation that is comparable to that of the Sanger. We also see an effect of telomere length, whereby short telomeres do less well than patients with longer telomeres. So finally, how do we combine all of this data together? At the moment, we're just getting marker after marker after marker, and it's not practical to go into the clinic and have an enormous list. What do we do with all of these things? Well, we've been looking at a technique called non-negative matrix factorization. So this is a technique that was developed for facial recognition software. And the idea of it is that you take an initial data frame, so in this case, your data would be a face, a photograph of a face. You then break it down into 2 non-negative matrices. The first matrix represents, in this instance, the facial features, so eyes, nose, mouth. The second matrix is the importance of those features. And when you multiply them back together, you're able to produce the individual face. So this allows us to produce 2 matrices that are able to capture and explain the underlying structure of that data, so the underlying structure of that face. So what we've been trying to do is to use this methodology to capture the underlying structure of our combined markers. So first we use Poised-Cox regression to identify in a trained way the features that are most significant in determining progression. And then later we take these features, be it mutation burden, telomere length, driver gene mutation, number of specifically mutated genes of interest, and we combine them with non-negative matrix factorization into prognostic groups of interest that explain the underlying complexity of the data. And these are the first results from this grouping. We can see that the TP53 deletion 17p abnormality group split off very nicely at the beginning. We also have 2 significant groupings that are in the middle. And interestingly, we've managed to produce a prognostic group that actually do significantly better than the IGHV hypermutated group. So in doing this, because this is a bioinformatics methodology, the idea behind it is that you can take a single VCF file and then program your software so that you could run through all of these steps, all of the marker identification, the non-negative matrix factorization, at just the click of the button. So you would be able to assign a patient grouping just that simply. So the takeaway messages are that we have the largest whole genome cohort of patients with CLL. Potential new gene targets found. Whole genome and non-coding analysis can determine factors related to progression, and combining data has determined 4 new risk groups for CLL. So I'd just quickly like to thank everybody in the SHU group, our CLL collaborators, the Streff group, the Halston group, and the patients, most importantly, and the clinicians who have gathered that data. And thank you for letting me speak.
Thank you, Kate. We have time for one question.
So here, I have a quick question. Yes. So regarding the genomic complexity definition, you're using the cutoff of equal or greater than 4 copy number aberrations.
Yeah.
So my question is, as you know, the cutoff used for calling those CNAs will greatly influence the number of CNAs you will call from a genome. So how do you define the cutoffs in your study? I mean, the size and the mosaicism level, et cetera.
Yeah, well, at the moment, we're trying to, so the idea of the grouping method is that it's powerful enough that it can sort of take in anything you can give it, really. So you can define it at 4, you can define it at 3, you can change the cutoff. It's all very flexible. And at the moment, we do actually have a different group working with us who are trying to determine the most optimal choice for genomic complexity in our data. And the idea is that then we can feed that into our models. So at the moment, the cutoff is still quite flexible. So 4 is what we've used at the moment, but we are in investigations to get a more accurate reading.